So, to add a wee bit more to knowledge, you've been through ultrafiltration. You should be able to tell me in that the full 5 parts of an ephron. So ultrafiltration is involved in the Bowman's capsule. There's frequently a part A CPQ in it, and then part B tends to be on this, called selective reabsorption. Now obviously reabsorption means take back, selective means only taking some things back. So what we're going to look at is the what, the why, the how. That's what is reabsorbed, why on earth you never want to do that, and then the marks in the exam really for how processes. Nothing new, they're all processes that tie back into AS. What's reabsorbed? Everything but urea. Why they're reabsorbed? Well, we need glucose for respiration, we need the amino acids to make proteins, we need the ions for balance, we need the water for maintain the water potential and hydration. We need the proteins in there as well. How? Well, that's you have to figure out, is it osmosis? Is it diffusion? Is it active uptake? Is it pinocytosis? So out of that, if you can get that all gathered together, we're doing a brilliant job moving forward. Thank you for your efforts. So here we have not very pretty start to an ephron, but it will do. Here, Bowman's capsule. Here's where the glomerulus would fit. And then coming out of it, we have what's called the proximal convoluted tubule. Okay, and I'll go into this as the descending limb of Liebowhelli, and away over here you'd have your ascending limb, and away not on the screen would distal and collecting duct. So we're just going to work at what goes on in this region. They are not very straight lines, but they'll do rightly. So you've explained ultrafiltration on the basis of size. So as a result of that, we can say that water and glucose and amino acids and ions and urea and small plasma proteins can all pass through due to the pressure and the fact that the basement membrane keeps out the large plasma proteins. This is what makes up the filtrate. So it's in here and it's about to go down along this bit. Now looking at that list, if I was to tick things that you really don't want to be losing too much of, well, you don't want to be using too much water, but we'll come to it again. You certainly don't want to be paying glucose out. You certainly don't want to be paying too many amino acids out. You might need to get rid of some ions. The whole purpose of this process is to get rid of that toxic waste. Remember, that is a nitrogenous compound made from excess amino acids. Certainly plasma proteins, small ones, you don't want to lose. They're very important for the osmotic balance of your plasma. So what we're going to see in simple terms is as the fluid, the filtrate, passes and moves along, remember it's being driven by the pressure. More and more is being forced in, so it's just pushing itself along. We will see things be reabsorbed out. Now, they have to go somewhere, and that's why in the previous video I showed you the efferent arteriole coming out of the glomerulus goes and tangles itself all around, in behind it, in front, going round and on, all around the proximal tubule. Now I'm going to remove that because it just makes my diagram look confusing, so do remember. And do remember, this has lost a lot of water, so this is very psi negative. So as soon as it starts to wrap around this proximal tubule, there is a movement of water by osmosis. There's an osmotic pull, and that water just goes back in to the vasa recta due to the water potential gradient. Now let's get rid of some of those lines. What a really annoying noise. So water, and along with the water actually ions will follow. Now water will move by osmosis, the ions will diffuse, and you're starting to pick up some things. Now glucose actually needs to be taken back. Remember, a sign of a diabetic would be there's too much glucose in your blood because of the insulin, and it doesn't all get reclaimed or reabsorbed. Glucose and amino acids go back by active uptake, which we know from AS1. We know it requires a carrier plus ATP, and the carrier is specific, so you need a specific carrier for glucose and a specific carrier for each amino acid. So we've got water back, and we've got glucose back, and we've got our amino acids back and our ions back. Let's see, small plasma proteins. Small plasma proteins, handwriting's getting worse. That is taken back by a form, remember endo and exocytosis, a form of endocytosis called pinocytosis. Abbreviates are sort of as like, abbreviates is a nonsense word. It's called cell drinking. Now remember, that's a type of endocytosis. So we've accounted for water and the ions that go with it, and we've accounted for glucose and amino acids, and we've accounted for the small proteins. Those are 5 marks, easily more than 5 marks when you describe the process. Urea we don't want back, therefore there's no actual carrier protein for it. However, a little urea does diffuse back. So a little urea will diffuse back. It just happens, but there's no active uptake of it. So frequently in the exam you'll write, there is no specific carrier for urea, therefore not actively reabsorbed. So if you imagine, here's our urea, lots of it have been left behind, but as it goes along and travels along here, more and more water is being reabsorbed. Now, what effect will that have on the concentration of urea? Well done, Oren, you actually got a question right. The concentration of urea increases, and in an exam they will ask you why it increases. All you have to say is due to the reabsorption of water. It's like me leaving a coffee pot on the coffee hot plate, water evaporates, the coffee towards the end of the day will taste stronger and stronger, simply because you got the same amount of solutes, urea, in a smaller volume of solvent, water, because you reclaimed that. Okay, I'll go through in another video the detail of this bit here, because we have to go through how is that wall specialised for the active uptake. So try and get your head around this as best you can.